Hi. Hello. Thank you very much for being here. My name is Santiago Montero-Mendieta, and yeah, today I'm going to talk about my PhD plan, which is entitled A Genomic View on the Diversification of Neotropical Frogs, and my main advisor is Carles here. So, as you all may know, the theory of evolution is based on the idea that all species are related and they gradually change over the time, and the accumulation of small changes can produce that some species get enough differentiation, some populations of the same species get enough differentiation that they will not be able to reproduce among with other populations of the species anymore. So when this happens, we say that it's speciation event, right? So what's speciation? So the speciation, yeah, it could be a very complicated topic, but I'm just going to say that it's the formation of a new and distinct species in the course of evolution. And these are some of the mechanisms. I'm not sure if there is more, but I think these are the main ones that can produce speciation in natural populations. So what I'm gonna, what I wanna say is that before speciation was mainly studied with using the speciation using genetic resources was mainly studied in model organisms such as human and mouse. However, with the high throughput sequencing techniques such as Illumina, now speciation is possible. You can study also speciation in non-model organisms, and that's mainly what we call speciation genomics. So here on this slide, I just wanted to say that before using Sanger sequencing, we had to select some specific markers, and we had to kind of believe that these markers were saying us the truth. But you know that it happens that sometimes the markers that you are using are not representing the species, the real species tree. So with genomics, we don't give a shit about that. We just we just sequence everything and we get everything, and then we can study things like we can find which are actually the genes that are involved in speciation, and we can find genes that are involved on hybridization, and also genes that are involved on adaptation, and many more things, but I'm just here saying the more important things for my thesis. So I'm going to use neotropical amphibians, and neotropical amphibians are the 50% of the amphibians in the world. Actually, there are many species that are being described every year, and some others that become extinct. But the thing is that there are few studies that have applied genomics to study the speciation processes in amphibians. And so still we don't know what are the processes that build this huge diversity of amphibians in the neotropical regions. So our study model are the frogs, a genus of frog called Oreobates, and this genus of frogs, it's nowadays composed by 24 species. And the good thing about these frogs, they are not very cool because they are all brown, and but the good thing is that they are distributed across a wide range of habitats and attitudes. So you can find these frogs living in cloud forest, montane forest, lowland forest, dry forest. They are all in South America, but very different habitats. So also there are some trees, but these trees are made only with 1, 2 genes, so I don't believe these trees that much. And what we want to do is to get the idea of what's going on here with these species. So also, we think that this is one of the best non-model species that we could ever dream, because there is species that have been described but by only one sample. Also, they are extremely difficult to find. You can ask Carles later about that. And what I was saying is that our collaborators here have been working with this species by the last 20 years. So we have access, like in blue you can see the species that our collaborators have described. So most of them have been described by our collaborators, and in green you can see the samples that we have available from our collaborators. So we have something that not many people can have in the world, I think, at least with this genus. So that's why we are using these frogs. So the research goals of my thesis, the main research goal is to study the evolution rates, demographic history, adaptation patterns, and on the frogs of the genus Oreobates. And I have subdivided this in, I'm not going to say four chapters, but 4 stages, because it can change. But the first stage would be to create a phylogenomics tree to study the relationships among Oreobates. The second would be to study evolutionary history, which means study variation in evolutionary rate. The third one would be the demographic history, in which we want to track demographic changes through time and the correspondence with habitat changes. And finally, it's about adaptation. We want to identify genes that have been differentiated between populations. I'm going to explain these 4 stages into more detail in the next slides. And then I will present some methods and results. So on the first stage, the phylogenomics, our idea is to build a highly supported tree with using genomics data, using genomic data. And with this tree, we will use it for downstream analysis, but also to solve some questions that can be solved with our phylogeny, which, for example, we know that there are species of Oreobates that inhabit the highlands and the lowlands. So we want to build a phylogenetic tree, and we want to know what's happening here. I mean, we want to. The thing is that it has been hypothesized that Oreobates was originated on the highlands. So that's a little bit weird, because normal species originate on the lowland, and then they go to the highlands to conquer new habitats. But yeah, in our case, we want to know what's happening here, how many species and how many colonization events. And yeah, we can solve that with the phylogenomics tree. So on the second stage, on the evolutionary history, we want to study the variation in the evolution rate of Oreobates. This previous studies on our group here from Álvaro have shown that in species that inhabit the highlands, the rate of evolution is lower, right? The evolution rate is lower. So we want to, but on the study of Álvaro, this was not very clear, I think, because sorry, because it was quite clear for mitochondrial genes, but not for nuclear ones. So we want to use genomics. We want to use all the genes. We want to know if this is true for all the genes. And so for that we will use genomics. So we want to know if for example, the evolution rate on the frogs that on the Oreobates frogs that inhabit the highlands is like slower than the frogs that inhabit the lowlands. The third stage, the demographic history. So we want to study the effect of the past environmental conditions on the Oreobates demography. So as you may know, the Andes, these frogs inhabit the Andes. Sorry, I don't know if I will tell you. And the Andes was originated about 20 to 30 million years ago. So these frogs, as they were originated because of that, it has been hypothesized that also they are from this time. The thing is that the frogs that inhabit the highlands, they are supposed that in the glacial cycle on the Pleistocene 2.5 million years ago, they had to survive somehow, and it's hypothesized that the populations were increasing and decreasing following the glacial cycles. So we want to test if that's true, if the species of Oreobates that inhabit the highlands have different populations, how do I say it? Yeah, well, you understand what I mean. If they have like different, if the population has been changing differently from through the time compared to the species living on the lowlands. And the fourth stage is the study of adaptation, in which we want to study if there are regions that can be different between a species because of adaptation. So, for example, this frog here, Oreobates ruralis, can be found in I think up to 3 or 4 different types of habitats. So, which includes like cloud forest, which is quite high, and then also mountain forest and dry forest. So we want to know if the different populations have different genomic regions on the genome that are different because of adaptation to environmental. So our initial idea was to use this kind of approach that, it's quite adapted from our collaborators in Sweden, which is because the problem is that we don't have a reference genome in frogs. So we wanted to build a transcriptome and then use it as a reference to do whole genome sequencing for the other species. And then for that we would create an exome assembly, which is the transcriptome. The transcriptome is the genes that are expressed, and the exome would be like the genes that are expressed and a little bit more. So the problem is that this study from our collaborators was quite good because it was on herring. So herring have small genome sizes. On frogs, the problem is the genome size is a problem because it's quite it can be quite big. So we thought that this could be important to look at the genome size, and we spent some time at the beginning of my PhD looking onto this. We tried to develop a method to estimate the genome size in amphibians through real-time PCR. This was not very was not a very conclusive study, so we but the thing, what I want to show with this slide is that on green here you can see that the amphibians have very large genome size variation on the genome size compared to the birds, reptiles, and mammals, which are also vertebrates. So that's quite a problem. So we decided to use, instead of the whole genome, we decided to use the transcriptome, which is like a reduced representation of the genome. So that's the methods how we are doing the studies and everything. So the problem also with transcriptome is that if you want to do transcriptome sequencing, you need to have the tissues stored in RNA later or a buffer that it's very good for keeping the tissue as it was at the beginning when it was collected. So the problem is that this is not always possible to in the field to do it because well, it requires some things. But anyway, the problem is that we only had like very few samples stored in RNA later. So we end up that we only could do the experiments with one sample from one species. So that's quite a problem. But we decided to keep going with this, and we use this approach that I'm showing you here, which it basically is that from one sample you sequence the transcriptome and you create a de novo transcriptome assembly. And after that, when we have the de novo transcriptome assembly, we use this to create a capture protocol experiment, which is something very novel, and I will explain it later in the last slide. But the thing is that this allows us to have the transcriptome for all the other species only from using only the transcriptome of one species at the beginning. So it's quite a good technique. And with that, we could solve, hopefully, all the questions that I was talking at the beginning. So I'm also showing you this here, which is the transcriptome workflow that I use to create the de novo transcriptome assembly. I'm not going to talk about this too much. I'm just showing this so you can make sure that I'm working. And this is, the thing is that we will publish something probably that will help other people to create the de novo transcriptome assembly. I think it could be quite useful. And yeah, here I'm just also showing you like some results. So at the beginning with this, with the sample, we started with a very large amount of data, but then we end up with a data set about 18,000 genes, and from that we created the experiment for the Exon-capture that I was talking before. So the thing is that, yeah, here I'm also showing that, yeah, there are some results. And this is results about the annotation. And the thing is that the transcriptome, the annotation, can be different depending on the data set that you are using. So we detected that from the 18,000 genes, there are 10,000 genes that are orthologous with Xenopus tropicalis, which is, of course, if you add more species, then the orthologous will be less. But that's the thing that you will use to build the phylogenies. So also here, this slide is some transcriptome results, which I was interested in know which are the functions of the categories, the gene ontology categories that are more common in our transcriptome. And you can see like most of the genes are involved on cellular processes, also single organism processes, binding, well, quite general functions. And here with this slide, that's the last about results. And I just wanted to show you that it really depends a lot also, again, on the database that you are using. In our case, we are working with a non-model species, so I had to use SwissProt, which includes a lot of different organisms. And as you can see, the main hits here are from Homo sapiens, and we are working with a frog. But that's quite common because, I mean, on the database, the most common thing is the Homo sapiens. So yeah, you will get the more hits depending on the database. So the last slide and what is next. So the next thing that we are going to do is what I was talking at the beginning. Well, actually, we are already working on this, but is to we built this de novo transcriptome assembly. Then we gave this to a company in the States, and they built for us something called capture proofs, and these are just DNA sequences that are complementary to our transcriptome of reference. So this means that we could use this molecular proof. This molecular proof will hybridize with the DNA of the other species of Oreobates, and with that we will be able to have the transcriptome for all the other species. And with that we will identify orthologous genes and test for the initial hypothesis that I was talking about. And yeah, thank you very much for your attention, and I will take any questions if you have any.